welcome back to The Explainer. So today we're diving into one of the absolute most fascinating detective stories in biology. Now, as focused learners, you already know that the final product of any living organism is incredibly complex. But how does that blueprint actually get drawn in the first place? Well, we're going to explore the genesis of geometry, specifically how a microscopic worm, C. elegans, establishes its entire three-dimensional body axes from absolute scratch. It really all starts with this fundamental biological riddle. Think about it for a second. You've got a single, perfectly round, seemingly completely featureless egg cell. How in the world does this microscopic little sphere know exactly where to put its head, its tail, its back, and its belly? I mean, how does it map out this intricate 3D geography before a single organ even exists?
Okay, so here's our end goal. We're looking at the fully formed adult Caenorhabditis elegans nematode. It's tiny, sure, but it is beautifully complex. We're talking a full nervous system, an intestine, musculature, a reproductive tract. It's got a distinct anterior mouth, a posterior tail, a dorsal back, a ventral belly, and of course distinct left and right sides. But like we just established, it starts entirely from one single cell. This journey from a total blank canvas to this highly structured adult relies on a rapid-fire sequence of mechanical forces and chemical conversations. And the trigger for this whole construction project, it's actually a super unusual fertilization event.
Because to build this worm, C. elegans completely throws out the standard biological rulebook. You're probably used to thinking of a typical sperm cell as being, streamlined with that long whip-like tail used for swimming. Not here. The sperm of C. elegans is completely different because most of these worms are actually hermaphrodites, meaning they produce both eggs and sperm within a single body. Fertilization happens internally when the egg rolls through a region called the spermatheca. And the sperm waiting there, they're small, they're round, and they have absolutely no flagella. Instead, they just crawl along using the slow amoeboid motion. So when one of these quirky, crawling sperm finally fuses with the egg, it isn't just delivering DNA, it's actually delivering the very first architectural command.
Section 1, the anterior-posterior turf war, dividing the one-cell zygote.
So that bizarre fertilization event kicks off our very first spatial challenge, where the single-cell zygote is rapidly divided into a distinct head-to-tail axis. What unfolds inside the cell is essentially a mechanical and chemical gang war between a family of molecules called PAR proteins. PAR stands for partitioning, and they literally do just that. They partition the cell. Before fertilization even happens, anterior PAR proteins are just floating everywhere in the outer layer, or the cortex of the cell, while the posterior PAR proteins are locked away inside the internal cytoplasm. But when the sperm enters, it brings along a structure called a centrosome. The microtubules from this centrosome touch the edge of the cell and basically create a safe zone. They protect a posterior protein called PAR2, which allows it, along with his partner PAR1, to sneak right into that outer cortex. Once PAR1 gets in, it goes straight on the offensive. It chemically tags or phosphorylates the anterior PAR3 protein. And that tag, it causes the cell's actin-myosin cytoskeleton to physically contract, literally sweeping those anterior PAR proteins away toward the completely opposite side of the cell. So the exact spot where the sperm entered is now fully conquered by the posterior proteins, permanently defining the tail end of the embryo.
And we can actually see this turf war frozen in time. Look at that bright green crescent localized exclusively to the right side. That's the PAR2 protein we were just talking about. The cell is physically dividing right down the middle, and because of that earlier sweeping contraction, the posterior PAR proteins are entirely cordoned off into one daughter cell, while the anterior ones are all trapped in the other. Literally, in a matter of minutes, the head-tail axis goes from being just a concept to being physically drawn out in glowing green geometry.
Section 2. Squeezing the dorsal-ventral axis. Mechanical forces at the 4-cell stage. Now, with the anterior-posterior axis totally locked in, the developing worm has to figure out its back from its belly, the dorsal-ventral axis. And fascinatingly, this doesn't start with chemical gradients. It starts with physical space. See, the embryo is trapped inside this rigid oval-shaped eggshell. At the 2-cell stage, the larger anterior cell, which is called the AB cell, gets ready to divide. And as it divides, it elongates so much that it actually becomes longer than the eggshell is wide. It literally has nowhere to go. It hits the curved walls of the shell, and the resulting mechanical squeezing physically forces the newly forming daughter cells to slide right past one another. One cell gets pushed toward the front, becoming the ABA cell, and one gets shoved toward the back, becoming the ABP cell. But here's the crucial part. This sliding action forces the ABP cell to ride up and wedge itself directly on top of another cell underneath it called the EMS cell. Just by running out of room, the ABP cell gets squeezed into a brand new physical location. And that new top position, That officially marks the future dorsal side or the back of the entire animal. This brilliantly illustrates how physical positioning sets the stage for destiny. Because the ABP cell got squeezed to the top, it's now the only cell physically touching a posterior cell called P2. And this simple touch kicks off a microscopic game of tag. The P2 cell carries a membrane protein called APX1, which acts like a signal. Meanwhile, the ABP cell carries a receptor protein called GLP1. Because they are literally jammed together now, APX1 reaches out and triggers GLP1. This contact-dependent chemical conversation breaks the cell's symmetry forever. It basically tells the ABP cell, hey, you are now fundamentally different from your sister cell. You are the dorsal surface. If this physical touch never happens, the dorsal-ventral axis completely fails to form. Section 3. The left-right twist. Establishing the final axis. Okay, so we've got a head and tail, we have a back and belly. Now we have to establish the final dimension, the left-right axis. I mean, how does a symmetrical bundle of cells know its left from its right? Well, establishing left-right chirality is kind of a slow burn that actually spans multiple stages. It goes all the way back to the single-cell zygote stage. Right before that first cell divides, the entire cellular mass undergoes a physical twist, a mechanical rotation of exactly 120 degrees inside its envelope. This really subtle twist creates an underlying chirality, a handedness, that pays off big time later on. By the 4 to 8 cell stage, the granddaughters of our original anterior cell naturally fall into asymmetrical positions, 2 on the left, 2 on the right. But the axis isn't fully biologically realized until the 12 cell stage. That's when a specific cell, the MS blastomere, reaches out and makes physical contact with just half of those anterior granddaughter cells. And just like we saw with the dorsal-ventral axis, this physical touch triggers an asymmetric signaling cascade, permanently distinguishing the right side of the body from the left. Actually, it's wild to think about, but this single touch at the 12-cell stage is what ultimately wires the left and right sides of the adult worm's brain differently. Section 4. Complex rules for simple worms. The illusion of simplicity. We've just traced the genesis of 3D geometry in C. elegans, right? From this amoeboid sperm entering a blank canvas, all the way to a 12-cell embryo that inherently knows exactly where every part of its future body belongs. It is just a stunning choreography of mechanical squeezing, precise protein gang wars, and physical touch. And the really crucial takeaway here is captured perfectly in this quote: even in an organism as simple as C. elegans, with only a few genes and cell types, the right side of the body is made in a different manner from the left. We tend to call these model organisms simple, mostly because they're easy to study. They have relatively few cells, they grow super fast, but as the research shows, their developmental networks are anything but simple. They are brilliantly complex machines that honestly just humble biologists time and time again. Which leads us to a final thought for you to ponder. We just spent this explainer unpacking the intense rapid-fire mechanics required to build a microscopic worm that is scarcely a millimeter long. So, if C. elegans relies on such deeply complex mechanical squeezes and chemical networks just to figure out its left from its right, what unimaginably vast, invisible choreographies are happening right now to build the rest of the living world? It really makes you look at every living thing, from a tiny snail to a human being, with a whole new level of awe. Keep learning, keep exploring, and we'll see you in the next explainer.
Thank you for watching. See you in the next one. Stay tuned.